On April 1, 2026, a cruise ship carrying 150 passengers and crew departed Argentina for a routine Antarctic expedition. Within 5 days, the isolated vessel functioned as a closed-system incubator for a lethal pathogen. The voyage began here, in the port city of Ushuaia. For the first 5 days, the ship's isolated environment remained stable. Then, a 70-year-old Dutch passenger developed what appeared to be a mild fever and headache. Within days, his respiratory system failed, and he died on board. Shortly after, the infection branched outward. His wife collapsed in transit, and others developed identical symptoms. By early May, the WHO confirmed a multinational crisis. Clinical data revealed rapid localized transmission and a case fatality rate stabilizing at nearly 50. Patients experienced a short incubation period with flu-like symptoms, which rapidly escalated into severe pulmonary edema, their lungs filled with fluid, culminating in systemic shock and asphyxiation. Tissue testing and lab diagnostics confirmed the pathogen was a strain of hantavirus. This diagnosis presented a significant biological problem. In virology, hantavirus is classified as an environmental zoonotic pathogen. To get infected, a human must inhale aerosolized droppings from a specific rodent vector. If hantavirus requires direct environmental exposure to spread, secondary infections in the middle of the ocean represent a clear contradiction of established transmission models. To understand the anomaly on the ship, we have to look at the baseline mechanics of the virus. Hantavirus is a broad global family of orthohantaviruses, where every individual strain is biologically tethered to a specific regional rodent species. Modern virology first documented this family in the 1950s during the Korean War. Over 3,000 soldiers stationed in field encampments fell ill to an unknown hemorrhagic agent. It took 30 years for researchers to isolate the virus from local field mice and identify the source. This map shows how the orthohantavirus family evolved. It split geographically, resulting in 2 distinct and highly lethal clinical syndromes. Eurasian strains cause hemorrhagic fever with renal syndrome. The virus attacks the vascular endothelium, causing internal bleeding and acute kidney failure. Conversely, strains in the Americas cause hantavirus pulmonary syndrome. The host's own extreme immune response triggers a massive fluid leak into the alveoli, effectively drowning the patient from the inside out. For both syndromes, there are no FDA-approved vaccines and no specific antiviral treatments. Doctors can only provide supportive care while the patient's immune system attempts to clear the pathogen. The baseline orthoantivirus is a highly lethal pathogen locked to its geography. Prior to the cruise ship incident, human infection required direct physical overlap with an infected rodent's habitat. This strict geographic limitation means that human outbreaks do not happen randomly. They are driven by environmental ecology. The mechanics of this zoonotic spillover are best demonstrated by an outbreak in the American Southwest. In May 1993, a cluster of young, healthy individuals in the Navajo Nation died rapidly from severe pulmonary edema. Epidemiologists traced the root cause back 2 years. A massive El Niño climate shift in 1991 altered weather patterns across the region, functioning as a primary environmental trigger. This graph illustrates the trophic cascade. The El Niño system brought unusually heavy rains, which in turn produced a bumper crop of piñón nuts, flooding the local ecosystem with food. With a sudden surplus of calories, the population of the local vector, the deer mouse, exploded. As millions of mice began competing for space and entering human structures, the concentration of infectious, aerosolized droppings reached a critical mass. The virus spilled over into human populations. Orthohantavirus outbreaks are predictable events tied to ecology. When specific climate conditions create a resource pulse for the right rodent vector, human infections follow. It is a misconception that hantavirus is an isolated, regional phenomenon. Data shows the threat is more widespread. Between 2005 and 2008, researchers conducted a seroprevalence study in the Vellore district of Tamil Nadu, India, searching for hantavirus-specific antibodies across different demographics. They found a 4% antibody presence in the general population. In high-risk groups, like warehouse workers or members of the Arula tribe who catch rats occupationally, that exposure rate was significantly higher, correlating with patients suffering from chronic kidney disease. Hantavirus is a globally endemic pathogen, circulating wherever humans and rodents overlap. Yet its spread has always been throttled by the fact that an infected human could not pass it to another human. On the cruise ship in April 2026, the Dutch couple infected at least 3 others on a vessel with 0 rodents, bypassing the standard rules of zoonotic transmission. Lab diagnostics isolated a specific variant, the Andes orthohantavirus, a strain first identified in southern Argentina in 1996. Out of the entire global orthohantavirus family, the Andes strain possesses a unique genetic mutation. It is the only known variant with confirmed human-to-human transmission capability. Contact tracers tracked the Dutch couple's movements prior to boarding. During a birdwatching excursion in Ushuaia, they visited an active landfill, an environment teeming with the local rodent vector. They inhaled the virus there and carried it onto the ship. Even so, the Andes strain operates under strict mechanical limits. It does not spread through the air like influenza or COVID-19. Transmission requires prolonged physical intimacy or direct exposure to infected bodily fluids. Furthermore, the virus is limited by a severe temporal window. Patients are only actively contagious for about 24 hours at the very onset of their fever. While close quarters allowed the index patient to pass the virus to his wife and caretakers, that contagious window rapidly snaps shut, preventing exponential spread. The Ushuaia outbreak occurred because an anomalous strain capable of human spread was introduced into a dense, closed population. However, the contagion was limited by its own narrow biological transmission window, which prevented the pathogen from moving indefinitely through the vessel's population. For global health officials, the immediate priority was evaluating the Andes strain for pandemic potential. To trigger a global pandemic, a pathogen requires 2 concurrent traits: high clinical severity and efficient epidemiological transmissibility. While hantavirus meets the criteria for severity, it lacks the transmissibility required for a global event. The absence of airborne human-to-human spread, combined with the microscopic contagious window, creates a natural biological firewall against dissemination. The actual threat remains localized environmental exposure, particularly as climate cycles trigger resource pulses and rodent population spikes. Because modern medicine lacks a cure, defense against orthohantavirus is strictly behavioral. Risk mitigation relies on rigorous environmental hygiene, sealing structural gaps against rodents, and utilizing respirators before entering enclosed high-risk storage spaces. Hantavirus is a permanent, silent fixture of global ecology, rather than a structural threat to global population stability. It is a deadly pathogen kept at bay not by modern medicine, but by understanding the mechanics of spillover and respecting the boundaries of